This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 779, recorded on July 9th, 2021. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today from Western Massachusetts, Alan Dove.
Good to be here. And I was just looking over at the weather, and the first thing I see on the Weather Service page is multiple hazards in effect, which is always a nice way to start off with that. Flash flood watch, hurricane local statement, flash flood warning. It's actually stopped raining at the moment, but it was pouring and probably going to start up again soon.
Yeah, we had all that yesterday here. We had enormous rain yesterday and through the night. And now the sun is out and it's 27 Celsius.
Also, wow. It's 19 C, 67 Fahrenheit here.
Yeah, when there's no sun, that's what happens, right?
Right.
Also joining us from Madison, New Jersey, Brianne Barker.
Hi. Unsurprisingly, warm, beautiful, sunny here, 84 Fahrenheit. Although if you ask my weather app, it would be a surprise because my weather app thinks it's quite cloudy, but my eyes don't agree.
Did you have pouring rain yesterday?
Oh, yes.
What was I recording? Yesterday was Thursday. I guess it was TWiV, and thunder and lightning, and it poured. And then when I left, I got soaked. I started taking the train, right? And then yesterday, they're like, we have 1 hour delays due to switch problems. You think trains would figure out how to get switches to work in the rain?
Well,
you'd think.
How much rain? I mean, everything's got a limit.
I suppose, but it turned out not to be so bad. Although then Amy said the subway flooded yesterday and she had to, anyway. Yeah, for trains that go underground, it's an issue, right?
Yep.
Mm-hmm.
Okay, so, and that I understand was Elsa, right? Disney princess hurricane,
yes.
Correct?
Yes, let it blow.
We have a public service announcement. There are 2 American Society for Virology sponsored town halls coming up this week. One on Monday, July 12th in Espanol, and one in English, Tuesday, July 13th, asv.org/education. Please encourage your friends and family to get their questions answered free, Zoom format with friendly, knowledgeable virologists. Does anyone speak Spanish, Brianne?
I'm not fluent in it, but I can probably read this if you want me to try.
Vamos. Or whatever.
Sí. ¿Tienes preguntas sobre COVID-19? Encuentro respuestas a tus preguntas con la ayuda de expertos en virología el 12 de julio a las 7 p.m. Central Time. Esta reunión será en español. Dile a tus amigos y danos un retweet.
Pretty good. You sound like one of those soccer announcers,
yes.
You know?
Oh, I can do that.
Yeah, but you need the horns blowing in the background.
Right.
All right, great. Yeah, I couldn't do that. If they did,
no Spanish for me.
You don't speak Spanish, Brianna?
No.
I somehow thought
French.
you did. French.
No,
Yes.
speak French.
Okay. All right, onwards to science. We have 2 snippets in a paper. So snippets, COVID, paper, not COVID, but very cool nonetheless. The 2 snippets have to do with the same thing. And this is a question a lot of people are asked. If I've been infected with SARS-CoV-2 and I've had COVID and I survived, and you wouldn't be asking the question if you hadn't, of course, do I need to get vaccinated? And these 2 papers address this and really provide a resounding yes and why too, right?
Yeah.
And they were both published in Science, I believe in the same issue, 372 and 372, yes, same volume. Yeah. And so the first is mRNA vaccination boosts cross-variant neutralizing antibodies elicited by SARS-CoV-2 infection. And this is
they're both in the same volume, but they're actually a few months apart. One of them came out in March and one came out in June.
It's always a risk that the volume is split over. Yeah.
Yes.
Okay, that's fine. But still similar experiments. In the first paper, we have, I don't know, the first author is Leonidas Stamatatos, and the last author is Andrew McGuire. Maybe there are two last authors, also M. Juliana McElrath. This comes from the Fred Hutch in Seattle, University of Washington, fondly known as UW, right? Is that UW or, yeah, I guess so.
Yeah, that's UW.
I think so, yeah.
We have University of Montreal in Canada, McGill University. And these are both similar studies where they take kind of small cohorts of people. Some of them have been naturally infected, and then they get revaccinated once and twice, and then some have never been infected and they just get vaccinated, and they compare. In this paper, they look at the neutralizing antibodies. In the other paper, they actually look at neutralizing antibodies and T-cell responses.
Yes, which was good to see.
Yes. Always makes me happy to see T-cells.
I thought, oh, Brianne will like this.
Yes, absolutely. I also have to say that I read the sort of HTML version of the this one that we're talking about right now.
Yeah.
And there's a little description from Science at the top, but right before the abstract. And the title of that is called Boosterism Could Save Lives.
That's cool.
Which I also
Boosterism.
thought was pretty funny.
Oh, that's a cool. Boosterism could be in our title, couldn't
Yes,
it?
it could.
Is that a word? It now is, right?
I think it is. It's in science, so it must be.
Yeah. It's usually used in politics or
Right.
related fields.
Now, before we dive into this first one, or dip our toes in because they're snippets, I guess. Dip our toes, yes. There's a sentence from the introduction, which I think is very interesting and important. Most serum neutralizing antibody responses elicited during natural infection are directed at the receptor binding domain of spike. Isn't that interesting?
Yes.
So receptor binding domain, right? Antibodies block attachment. And, I always teach students, there's more than one way to neutralize. It's not just blocking attachment, but here, apparently not. For this virus, that's the way they work. So keep that in mind because that'll kind of be relevant here. And the real issue is here, they want to say, if we take an ancestral variant, like the original HU1 isolate, and compare it to a variant, the more recent variant, does any of this make a difference, whether you're infected and boosted or just boosted twice, et cetera? That's the idea. So they have two groups of people. They have 15 donors, and the numbers are small, but these are tough studies to do, so we're not going to get thousands and thousands of people. They have 15 donors who have previously confirmed infection. So they're called...
Was this healthcare workers or is that the other one?
I think...
the other one's healthcare workers.
Yeah, this is not healthcare workers.
Okay.
They just talk about vaccinated individuals.
Yeah, these are people. These are just us.
Whatever sample you can get. Yeah.
All right, previously confirmed. These are previously infected PIDs. And then they subsequently get one or two immunizations with mRNA vaccine. I think they used both mRNA's vaccine. I don't remember. mRNA vaccine. We'll get to that. And then they have naive donors who were not infected but got mRNA vaccine, 2 doses of these vaccines, okay? PIDs and NDs. So they take serum from these people and they use pseudoviruses to do neutralization assays. And they have pseudoviruses with either the ancestral spike or the B1.351 spike. So this is an older paper, so it has the ancestral nomenclature.
I think that is Delta?
Well, 351, no,
beta.
That's not
I think it's
Delta,
beta.
that's beta. I think
it's
and 117 is alpha.
Yeah, that's right. 117 is alpha.
Okay.
So the interesting part is that, all right, so the PIDs, 3 of them had asymptomatic infection. And 2 of them had no anti-RBD antibodies before immunization with the vaccine. So even though they recovered, they didn't have antibodies, 2 out of the 13 or whatever it is. So this is, that can happen. People don't respond. And in the 13 PIDs, the others who had antibodies after their infection but before vaccination, a single vaccine dose boosted these titers 500-fold. And across all of the PIDs, 200-fold increase in median RBD binding IgA titers after vaccination. So you get a really good boost if you've been infected and recovered by a single mRNA vaccine immunization. And they also looked at memory B cells, and spike-specific memory B cells can be detected in not all the patients, but some of them, right? Some of them had no memory B cells, and that's interesting because those are the ones that don't really respond as well to the vaccine immunization.
Right, and that might be a reason why we might want some of these people to actually get both doses of the vaccine to make sure they induce a good memory B cell response.
That's right. All right, so then neutralization assays. 12 of the 15 PIDs sera before vaccination, they neutralized the HU1 variant, the original. And the 3 asymptomatic patients had no neutralization activity in their serum, either low or they had low or undetectable anti-RBD titers and no neutralization. And also, of course, pre-vaccine sera from the NDs were non-neutralizing, as you would expect. And then the PIDs, after the first mRNA vaccine, 1,000-fold boost in neutralization titers. But the second had no effect. Second boost. So I guess you don't need that second one.
But if you had an asymptomatic infection and you don't know how,
yes,
right.
If you were symptomatic, you don't need it. But yeah, so you might as well just give everyone
go ahead
two.
And get the second dose.
Yeah, it's probably right. In the NDs, those are naive donors, 2 doses of vaccine gave titers that were 10 and 5-fold lower than those in the PIDs with either 1 or 2 doses. Isn't that interesting?
Right.
Mm-hmm.
So kind of a hybrid scenario. Natural infection immunization is really good. So
assuming you survive the natural infection and don't have any long-term sequelae.
Issues, yeah.
I mean, we don't encourage people to go get infected.
No. You do not want to get this virus. But if you have had it, you should still get immunized because you're going to have great immunity coming out of that.
Yeah, because people have said, why do I need to get immunized? This is it. You get a really great response. Yeah. And as we're going to see in a moment, it includes neutralizing the variants. So they said, summarize all this in PIDs who have memory to RBD, which means they're symptomatic. A single dose gives you a great memory response, and that is superior to a 2-dose regimen in uninfected donors. So, this is 15 people. What do we think? Is that going to be generalizable to the general population? If you looked at 100 people or 1,000, do you think it would be the same? I just don't know. So
there are other similar studies, which they cite here and that have been done, that have kind of come to the same conclusion that 1 dose of vaccine after infection does seem to give people a very high response. But I think it's important to note, in that sentence that you were talking about, that you just read, it is talking about PIDs who generate adequate immunological memory.
Yeah.
And that involves looking at memory cells, not just antibody titers, when they're making that conclusion. And so if someone has been infected, they might know something about whether or not they have antibodies in a yes or no kind of way, but they are not getting their number of memory cells checked at the doctor's office. That's not something that is done clinically at all. And so you don't know if you are this infected person who generated adequate memory or not. And so maybe 1 dose would be okay if you happen to already have generated memory, but we can't tell that, which is why that second dose could be really important. But I do think that in the people with memory from this study and from some others, the single dose does seem to make sense.
Okay. Yeah. Okay.
And I think this and the other paper together bring up an interesting future scenario where we're going to have a lot of people vaccinated. The virus will still be circulating. Variants of it will still be circulating because we're not getting rid of it. And you talked a little bit with Ron Fouché about this. Great episode, by the way, last episode. We'll have the virus going around. And so those of us who are fully vaccinated but never had it originally are going to get exposed. We may actually harbor an infection. It's extremely unlikely we'll end up in the hospital, though, as we're seeing, these vaccines are highly effective against that. But that should provide an immunological boost. And so it would be the reverse of these folks who had the virus and then got the vaccine.
Yeah.
You'd have the people who got the vaccine and then had the virus, and that's going to provide ongoing boosting. And I really do think that we're getting to a scenario where this will be our 5th common cold coronavirus.
Right. I, in fact, had a conversation with someone who is not an immunologist this morning and introduced them to the idea that not only is there such a thing as a secondary response, the second time you get an infection or the second time you get a vaccination, there are also tertiary responses and quaternary responses that can improve with each subsequent exposure. And so, as we go about and live our lives after having been vaccinated, we might get infections that might give us those tertiaries or quaternaries, but still not get sick.
I when I teach about immune memory, I always use the island example. That what are they called? The
Faroe Islands.
Faroe Islands, where memory is maintained for many years in the absence of re-exposure. However, it gets better if you get re-exposure, for sure.
Right. Yeah. And I use that example, and I also kind of use the example of different cohorts and their exposure to, say, measles, or their exposure usually to chickenpox.
Yeah.
How, someone who is perhaps a little bit older may have had chickenpox and had multiple additional exposures to boost their immune response. They may be less likely to get shingles when their immune response wanes, where someone my age who got chickenpox and then everybody younger than me was vaccinated.
Yeah.
So I've never really been around another person with chickenpox. I've never gotten those boosts.
That's right.
Means that I will very likely get shingles at some point.
Yeah. I mean, the vaccination does reduce circulation, but it's not zero.
Right.
And certainly because only polio is about to be eradicated right now.
Right.
These other viruses. But as Fouché said, flu, everybody gets challenged every year, and it boosts your immunity. So now they look at neutralization of variants. So they do B1.351, which earlier in the paper they had examined with monoclonals and shown that some of these do not neutralize infectivity with these variants. But before vaccination, 5 of 15 sera from PIDs neutralized B1.351, but only 3 had titers, 50% titers above 100. 7 of the 15 neutralized another variant of B1.351 with a deletion, but only 1 had titers above 100, and only 2 pre-vaccine PID sera had 80% neutralization of B1.351. Only 1 achieved 80% of the B1.351 with the deletion. So these recovered patients don't have reduced neutralization against B1.351, although, as Fouché said, who knows, it may be enough still to protect you, right? Even though it's reduced. The median 50% infectious dose of the pre-vaccine sera against the original variant was higher than against these two 1.351 variants. A single immunization boosted neutralization titers against all 3 variants and SARS-CoV-1. They also checked that in 13 of the 15 PIDs, although the titers were lower, 3-fold against 1.351, 10-fold lower against the 1.351 with the deletion. So that's one of the advantages of getting immunized after recovering. You get a nice response to these variants, a stronger response than you had originally, right? Okay, one more thing is it's interesting. So they wanted to confirm that it's actually antibodies against the receptor binding domain that's mediating this neutralization activity. So they actually deplete the RBD-specific antibodies from the serum of 10 PIDs and after 1 vaccination, and from 9 NDs after 2 vaccination. And that depletion gets rid of the neutralization. So it confirms that most of the neutralization is by antibodies against the receptor binding domain. So there you go. And in fact, both Pfizer, BioNTech, and Moderna vaccines were used in this study. So 2 doses of either one, you get neutralizing antibodies against the original variant, lower titers against the variant, and the PIDs, you get better titer against the variant after a boost from that, after a natural infection. So they say as long as RBD-specific memory B cells are induced, then a single immunization gives you a good memory response that boosts titers against the original virus and also against the variants, although at lower titers, which makes sense, right? It's fundamentally the same virus, which is it's kind of it's a memory response. So they say this is also important. In most of the previously infected vaccines, the anti-B1351 deletion titers were comparable to those against the vaccine-matched HU1 in uninfected vaccinees. And they say this is important because these titers were associated with 95% protection in the phase 3 trials. So we do have some idea that these titers, albeit reduced, are still going to be protective, which I think is really important.
Yeah. And this is consistent with what we're seeing in the epidemiology, that these variants are circulating. Looking at the US example, we actually have, we just passed the 50% mark for Delta. Mm-hmm. So there is apparently some kind of fitness advantage there. But almost everybody who ends up in the hospital with that or with the previous variant is unvaccinated.
That's right.
I mean, it is exceedingly rare for a fully vaccinated person to end up with serious illness with either the original or any subsequent variant.
In fact, where there are outbreaks in the U.S. and the States, there's low vaccination rates.
Areas with low vaccination rates. In fact, there was just a paper that came out mapping that down to the county level, and it's they've got, I think, 5 big dots on the map, and it's across mostly the southeastern U.S. I've seen a lot of news lately about Missouri and Arkansas, but there are pockets elsewhere.
I get called all the time. I just spoke to someone on CNN. What can we do? I said, get vaccinated. That's it. It's an easy solution. It protects against disease. What the heck? Yep. Okay, so that's that one. And the second one, a similar study, except they expanded to include T cells, and this is in healthcare workers, right?
Right.
And this was done in the UK, where they initially gave a B&T vaccine and then boosted 12 weeks later, remember?
Mm-hmm.
Yep.
They waited a while because they wanted to get more people with that original dose. And they looked at neutralization responses. And they had similar groups, right? They have people who were infected and recovered and then vaccinated, and then naive people who were just vaccinated. And the antibody responses are similar, right, to the other one. You get responses again. And they also look at B117 and B1.351. And again, if you were recovered and immunized, you get a nice response against not only the ancestral variant, but also the variants as well. But they also looked at T-cell responses because they say, this N501Y change in the spike, which is involved in ACE2 binding, some people think may affect T-cell responses. And so I guess the approach is to make is to examine peptides that are known T-cell epitopes. What they found is, depending on the HLA type, sometimes the changes can make a difference, which is really interesting, right? And in fact, this particular HLA DRB10401, they have transgenic mice that produce that HLA, and they find that the responses, T-cell responses to the peptide pool from B117 are reduced.
Right.
Which is interesting, right? Because in all the T-cell studies that I've seen, they don't look at HLA. They just take lymphocytes from a variety of people and throw the peptides in, and you don't see much of a difference. But I guess in certain people it could matter. So Brianne,
yes,
like this particular HLA, I mean, how many people in the U.S. would have this? Do you have a rough idea?
That I have no idea on that, DR. There are big variations between sort of the common and the rare different HLA types. And I don't know this one in terms of whether it's common or rare.
There are a lot of different types, right?
There are. And so I think that that's kind of the important thing about this T-cell response, both in terms of what we might predict and exactly what we're seeing here. All of this kind of makes sense to me with basic immunology. So if people were watching closely, when you mentioned that some people think that N501Y could change T-cell responses, I made a little bit of a face, I think. And the reason is that every person has a different group of HLA molecules. So they are the genes with the largest number of alleles in the human genome. So basically every single one of us has a different set of 6 MHC class 1 HLAs and MHC class 2s. And so it is entirely possible that a variant could mutate in such a way that it would change a peptide that would make a T-cell response in me less good if I happen to have this DR type. But that's actually not what the virus really cares about, because the virus, after it's in me, is going to spread to Alan. And if it's incredibly unlikely that the same change in me that gave it an advantage in evading T cells will be able to evade using Alan's MHC types. And so on a population level, we don't really expect there to be any particular variant that's going to change T-cell responses to everybody. And so we shouldn't see the same kind of selection pressure that we see with the antibody responses, where everybody has the same antibody epitope.
It's only in a persistent infection like HIV or Hep C.
I was just about to say, what you could see is if the virus is in the same person for a long period of time, there's a selective pressure for it to adapt to that HLA haplotype. But as Brianne said, that's not going to do it any good when it gets to the next host.
Right. And that's actually been shown, is that even in those persistent infection cases where you see the virus adapt to the host's HLA type, then if you look at transmission into someone with a different HLA type, it's no longer the most fit virus. And then we'll have to readapt.
But that's, that's consistent with what people have hypothesized about the development of these variants, is that they are more likely to arise in somebody who ends up with a long-term infection. Obviously, the virus has more time to kind of hang out and mutate. And then if they have acquired maybe something some changes that adapted specifically to that individual, but also some changes that maybe helped evade more general responses that everybody's going to mount against receptor binding domain, for example, then we may see a fitness advantage in that virus, and it spreads to a wider population.
But in an individual patient, they say HLA polymorphisms could be determinants of responder and non-responder status. So in a person, it could account for more severe disease in a particular person.
Absolutely.
Right?
So, my HLA response might be such, or my HLA types might be such that I make kind of meh T-cell responses. And so I have slightly worse disease because my T-cells aren't responding as well as yours.
Got it. So they say basically the case has been made that reductions in antibody neutralization of spike may be mitigated by protective T cells. Our evidence says after one dose, T cell immunity is mostly variably low, but also relatively unperturbed by the N501Y in general, except if you look at a very specific HLA, then you see a difference. But in general, it's not. A couple of other things that are worth mentioning here. Here's another sentence I highlighted. While much has been written about the impact of rapidly waning serum antibodies, our findings confirm that memory B cells are nevertheless primed and able to contribute a rapid, large response. And this is something we've been mentioning over and over. It doesn't matter if the antibodies wane. They're supposed to. You don't want them to be high, as Brianna said. And you have a memory response that's going to help you. And it won't help you immediately, so it's not going to prevent infection, but it will kick in really quickly and prevent disease. So I like that. That's very good. Here's the other thing that I thought is good. the rather large effect on B cell priming and restimulation relative to T cells may reflect the fact that among the nuanced differences between the licensed vaccines, the mRNA adjuvant appears to skew immunity to high neutralizing antibody titers, which may underpin its high efficacy. That's really interesting because the mRNA vaccines are fabulous, right?
Yes.
And maybe that's it, this mRNA adjuvant effect. It's very cool. I'm sure we'll see more about that coming. We just did a paper on TWiV yesterday, or Wednesday, a malaria mRNA vaccine. They put the circumsporozoid antigen in an mRNA and tested it in mice and some protection. It's really cool.
Okay.
It's very cool. All right. So both of these, the conclusion is if you had infection, you should get immunized. And
if you haven't had an infection,
get both
you should
doses.
get immunized.
You should get immunized, and they're going to give you reasonably protective levels of antibodies against the variants as well. And of course, what we really want to know is disease prevention, and the studies are suggesting that yes, the vaccines prevent moderate to severe disease with all of the variants. So the bottom line is, folks, please get vaccinated.
Yes, please get your vaccine.
Yes, the bottom line is vaccines are good.
Yes.
Vaccines help everyone.
Vaccines in general, and these vaccines in particular have been pretty spectacular.
Yet people want to make excuses for not getting them, right?
Yeah.
Right.
It's not approved, right? Oh, come on. It's experimental.
We went through this. We had Paul Offit on and discussed the entire approval process and what's different about an emergency use and how it's the same process with the same requirements. And the only difference is that you don't have efficacy data beyond a couple of months. But that's irrelevant because you don't see complications arising beyond a couple of months in any vaccine.
So I think that there's a really important point there. So there's a complications issue. And we don't see the complications after a few months. But the difference is, in fact, as you said, that we don't have the efficacy data.
Yes.
And so when people ask questions about waning immunity or how long this will last, that is something that we don't have because of the emergency use.
Yes.
As opposed to the full authorization. So that is a place where there is a difference. And if people have a question about what's different, knowing how long it will last is part of the answer to that.
That is an excellent point, yes.
But we do know that it works.
Yes. And we do know now that it lasts at least several months because people who got vaccinated at the very beginning when the vaccine first came out, autumn of 2020, still are neutralizing virus.
I mean, the worst that can happen is you need to take another one in 6 months, but probably that's not even going to happen.
I don't think that's
in
even
the
going to be an
meantime,
issue.
your life is saved. So what is wrong with that, right? I don't understand. But, we do discuss this a lot, but, not everyone listens to us. So it's a difficulty. I was on an NPR program the other day where someone said, well, maybe by the end of the year, the licensure will happen. Will that make a difference? I think people who don't want to get vaccinated will make other excuses not to get vaccinated. In fact, right?
There are polls indicating that some people say, "Oh, when it gets full approval, I'll get it." But I agree with Vincent that that's probably just the excuse they're currently using, and they don't want to admit to the poll taker that their real reason is because they think Trump won the election and the whole thing's a conspiracy.
Yeah, indeed.
Realistically, I think the take-home message from all of these papers regarding vaccines is exactly what science said at the beginning. Boosterism could save lives.
Yes.
Boosterism. This second paper, I forgot to tell you where it is from. It's from a variety of institutions in the UK, Imperial College, Queen Mary University, Public Health England, et cetera, et cetera. And the first author is Catherine Reynolds, and the last is Rosemary Boyton, and many people in between, including the UK COVID Sortium Immune Correlates Network. There you go. These are very good. And as Brian said, there have been others doing similar work, but these caught my eye and important questions. You can take a look at them. I think they're probably open access, right?
They are.
Yes.
And, good immunology in both of them. Good information that I think explains the answer to questions that people have been asking.
Yes. I mean, we always said, oh, you should get immunized even if you've been infected because we don't know the nature. But now we know exactly what it does. Here you go, folks. Get immunized, please. All right, well, our paper today, now we move away from COVID, but please stay because this is pretty cool stuff.
This is really cool.
Yes, it is.
Another science paper. This one titled Evolution of a Virus-like Architecture and Packaging Mechanism in a Repurposed Bacterial Protein. And this is first author Stefan Tedder, last author Donald Hilvert.
So I think there are 3 co-first authors.
Yeah.
Stefan Tedder, Nahiro Terasaka, and Angela Steinauer.
Mm-hmm. Okay. And this is from ETH Zurich, University of York, University of Leeds. And this addresses the issue of the early evolution of viruses. So there's one idea is that, and if you listen to the Eugene Koonin episode, you'll get more on that. One idea is that, early in Earth, self-replicating molecules emerged, self-replicating RNAs eventually. In the absence of protein, that was the RNA world. And then, eventually, and I'm going to make a big leap here, protein evolved, and then enzymes, and then DNA, and then cells. And then once the cells appeared, which is many
Vincent
years.
just fast forwarded through about 2 billion years, I think.
Yes.
There are many interesting questions.
That's the time-lapse version.
In the Koonin episode, people who want the fast forward version, there you go.
Yeah.
We talked about that. But the idea is that these replicators, which were, these are the primordial viruses, then they went into cells, probably initially accidentally, right? There's no directed evolution there. And there they stole capsids in the forms of proteins that could bind RNA. I think one of the first protein motifs thought to have evolved was an RNA binding motif. And so somehow these RNA binding proteins evolved into capsids to protect the RNA, which could then leave the cell. And that's a big leap there, going from a protein binding the RNA to a capsid that will protect it and package it specifically and nothing else. That's a lot of evolution. And that's what they're trying to get at here. How did that happen? And actually, spectacularly, they really make a lot of progress. It's really remarkable. Yeah. I mean, I was not aware of this going on, and I saw this and I said, wow, this is very cool. So what they do is they actually cheat a little bit where they're starting, because at some point they have to go back even farther. But there's an enzyme in a bacterium, Equifex aeolicus. The enzyme is called lumazine synthase. And this is an enzyme that naturally forms 60 subunit particles, which they call nanocontainers. Many enzymes do this, but it has no affinity for nucleic acids, right? And so they wanted to evolve this into a capsid of some kind. And there's previous work to this paper where they actually took this enzyme, AALS, and they redesigned it. First, they did circular permutation, which means they moved the amino acids around, leaving the structure of the protein the same, but changing the order of amino acids. And they added a peptide sequence which binds specifically to an RNA stem loop. The peptide is called lambda N plus, and the stem loop is called box B. So they stuck onto the end of this enzyme this peptide sequence that will bind an RNA stem loop. So you can see where this is going. They're trying to package an RNA by having these two things interact, right? And they published this previously. This variant was called NC1, nucleocapsid 1. And they did directed evolution in the laboratory. They got NC2 and NC3. This is cool. They selected for variants that would capture the mRNA for the protein of this nanocontainer with the stem loops at either end, right? So they put the mRNA encoding the enzyme, they flanked it with these stem loops, and then they selected for variants that would bind and encapsulate it. And they got it. They got NC2 and they got NC3, but the packaging of the genome was not great. Only 1 in 8 of these NC3s actually packaged the full-length RNA, but still, that's pretty damn good that they got to that point at all, right?
Yeah, I mean, you have to think about all of the potential mutations in genes that lead to these changes in the protein and how fortuitous it would be to find one that, A, can distinguish this RNA from all the other RNAs, just in terms of what it encodes, and also the full-length version compared to the non-full length version. And so, those are some pretty fortunate changes, but then an RNA that a cell with RNAs that have those changes will really have a selective advantage because that RNA can propagate.
That's right. Yep. I mean, they're cheating, right? They start with a protein that makes a container, and then they stick on a peptide that's going to bind an RNA, and they put the RNA on the mRNA but you can't. You have to start somewhere.
You can't wait 2 billion years.
You can't. And
your grant renewal is before
I presume in the future they can go back further to a single peptide or protein and try to do this. But as you will see, they get amazingly to where they want to go. So now they have NC2, NC3, and they want to make it better. So they do error-prone PCR of the coding sequence for this, where you get lots of changes in the mRNA. And then you can make the protein, purify it, and what they do is what they call nuclease challenge. And then whatever is left after nuclease challenge, they amplify and repeat this whole thing over again. And nuclease challenge, they have 3 different nucleases that are going to chew up the RNA of different sizes. First, they start with a 60 kilodalton one, which
was so clever.
Benzoase, then a 14 kilodalton RNase A, and then a 11 kilodalton RNase T1. So smaller.
Right. So
smaller,
the idea is the smaller RNase would be able to get into smaller pores, and you'd have to have a better capsid to protect against it. And so they start with a bigger RNase. If you're reasonably encapsulated, you'll probably escape that. But then we're going to up the ante. We're going to make it harder and make a smaller RNase. Okay, maybe you could get past the first one, but can you get past this little one? And then they go to the smallest one and they select down, and they do this sequentially because probably if you went straight to the smallest one, you just get no survivors.
Right. And they also do it over different time points to see how much, to give the RNases or the nucleases different amounts of time to actually try to access that RNA.
So when you think of what they're doing, they're taking they have the RNA in a bacterium. It's going to be translated to make the protein. It's going to assemble a capsid. They're going to purify the capsids, digest them with the RNase, and then what's left, they extract the RNA and do it over again. So that selects for assembly, right? Assembly of the capsid. It selects for protecting the mRNA from nucleases, and it selects for full-length mRNA because you take it and you put it in cells and you have to get the protein again. It has to be full-length in order to do that. And they have these...
And even though this is an accelerated process and they're using an artificial system, it mimics what happens in an evolutionary context in any ecosystem.
Right.
Because things don't, other than an asteroid hitting Earth and wiping out the dinosaurs, things don't radically change in all of the life around you at once. Your competitor gets a little bit better, and so you need to get a little bit better, and so your competitor gets, and there's this ongoing thing, and so they're mimicking that by starting with, okay, here's an RNase that can maybe get to you, and here's one that can more likely get to you, and here's one that can more likely, and going through rounds and rounds of it.
And they mimic it by making the changes by sort of random changes in the nucleic acid, which is what happens normally too. Yeah. It's not that you and your competitor are getting better and better because you feel like it. It's because you happen to have picked up a random change in the nucleic acid. And here, they're just throwing random changes in nucleic acid but sort of speeding up the process of picking out who's the fittest.
Right.
And allowing them to win.
It's all about fitness, isn't it?
Yep.
Yes.
And this is better than an approach where you try to design something to escape that, because that doesn't really get at the question, how did these originate? Here, by using this random element and letting the sequences figure out what's what, you're really selecting for the solution that evolution might arrive at.
And each time
So it's evolutionary boosterism.
Evolutionary boosterism. Oh, that's a cool title. Each time they do this, of course, they're selecting for changes that allow these things to happen that we just talked about, RNA stability and so forth, RNase resistance. So then they end up with NC4, nucleocapsid 4. And this paper basically compares NC1, 2, and 4 with each other. And during this evolution, in the beginning, the particles are kind of heterogeneous, and now NC4 is pretty much homogeneous, good protein yields, good nuclease resistance. They say NC1 the RNA is almost completely degraded with RNase A, whereas NC2 gets a little better, and 3 and 4, most of the RNA is protected from both nucleases. And again, this is just by these serial selections. 4 packages its own mRNA with really good specificity. I think it's 863 nucleotides long, and something like 80%, 87% specificity. So there's still some cellular RNAs packaged. I think they're ribosomal RNAs, but it mostly packaging its own, and about 2.5 full-length mRNAs per capsid, which means it's big enough that you could put a longer mRNA if you needed to, something in addition to the coding sequence. Maybe a polymerase or something.
Yeah.
Maybe something could evolve there.
That would be cool.
So Vincent, I have a question for you. How do you feel about that 87% efficiency compared to what we see in sort of other viruses? I mean, there are some viruses that have a pretty low particle PFU ratio. Yeah.
I mean, I think that's pretty good, but it's 87 plus or minus 19%. So some of them it's a big error bar there. But clearly some viruses do package other nucleic acids, and sometimes they have some roles, and others do not. So they're selecting for it here, right? So that's what they're getting. You get what you select for, but not always what you want. Is that how it goes? It's the same.
Yeah, but I feel like they're getting in the range of what you might see, at least in some viruses.
These capsids, as they get better and better, they have different structural changes.
This is so cool.
Yes, it is.
Anytime there's a structure thing, I go straight to the pictures first, and then look at the data. And I looked at the pictures. That's a virus.
Exactly.
That's an RNA. You made an RNA.
No,
virus.
They do cryo-EM, and you see these things are evolving towards uniform 30 nanometer diameter particles, and they say, in fact, the bigger ones can now put the whole mRNA in, whereas the smaller ones, the cavity inside was too small, right, to package the RNA. But the selection they used, depending on getting the mRNA in, it worked.
Yep.
And these are built, the NC4 is built with icosahedral symmetry. This is what stunned me the most, because I teach students that an icosahedron is the best way to make a very stable particle with the smallest number of subunits. You can do 60 copies of a single protein will give you a very stable particle, and that's done by icosahedral symmetry. And none of the other platonic solids are used for that. It's this one, and that's what they got. They got friggin' icosahedral symmetry. Isn't that amazing?
It all arrives at the same solution. You apply the same selective pressures, and you end up at the same endpoint. And it fits so nicely with what we see of viruses. You look at RNA viruses.
Yeah.
What shape are they?
Yeah. So
This is what you get.
they compare all of these, and there are big changes that occur. You know, the smaller ones have more heterogeneity, but they are still unusual, right? The UNC1 is what they say is an unprecedented 120 subunit protein cage, right? Remember, they started with 60 subunits, and now they're NC1, and these are made of pentamers mainly, which you would expect from that number. NC2 also, it's not homogeneous, but this one has 180 subunits, and they're getting bigger. And I always tell students, this was a teaching paper for me. So, you know, we have small viruses and bigger ones. How do we make bigger viruses? The proteins don't get bigger. We add more proteins.
Yep.
And here we go. NC2 is bigger, and it's got 180 subunits versus NC1, which had 120. And the cool thing is when you add more subunits, when you add more than 60 subunits to a capsid, when you go to 120 and 180 and 240, in the 60, you have all pentamers. They're in groups of 5. But as soon as you go above 60, you now have pentamers and hexamers, and that's exactly what they see here. That this is just amazing. They see hexamers and pentamers. And in fact, the final one, the NC4, 240 subunits with very small pores, a big interior that can fit a couple of these mRNAs. And they say this is typical of a T=4 capsid, where you would have 4. So the icosahedron is made up of 12 equilateral triangles. And you can further subdivide the triangles into smaller triangles. And that's what the T number tells you. And so this one would have 4 smaller triangles for triangular face. And the T number tells you the total number of subunits in a particle. So this is T=4. So the T number times 60 gives you the total number of subunits. And this one has 240 subunits, exactly as you would predict. It has pentamers and it has hexamers. The real interesting part is what allows this, and they can actually pinpoint the changes in the proteins that allow these kinds of interactions to put more subunits in. For example, one of the changes makes the end of one of the proteins flexible so that it can interact with the neighbor and make the particle bigger and more stable, right? Slight variations here and there, which they can see in their structures, repurposing of parts of the protein. And that's what happens when you select. These things happen. Now, this NC4 has very good nuclease resistance, but they think it's not just the protein. They think the presence of the RNA helps the capsid be more stable. So they make a variant of NC4 without the RNA binding peptide. Remember, they introduced into the protein a peptide that binds RNA, right, to get it packaged. And they take that out, so there's no RNA in there. And these are now less stable and heterogeneous without the RNA. So they think the RNA is playing a role in the assembly, which is something that we think about for viruses too. And this is great evidence for it, right? All right, so the other question is, remember, they put a packaging sequence in. They put in this peptide that binds the stem loop. And they said, but why is NC4 so much better at packaging than NC1? Turns out that the packaging sequence has been repeated like 37 times throughout the genome. And they actually do some what they call X-ray footprinting. They can see the protein next to the RNA. And it looks like these extra duplicated packaging signals are really involved in getting inside the capsid and making NC4 more stable. So that's pretty cool. There's 7 potential packing signals which arose during their evolution. And apparently some of those have been utilized. So a lot of data in this paper, actually. It's very cool. And in fact, I should say that they can see organization of the RNA in the capsid and contacts with the capsid protein, which we're just beginning to see in another number of other viruses. So this NC4 evolved a key element of RNA packaging.
Yeah.
It's just so cool. That is the story here. And so basically it tells you, and as Alan said, convergence of structural properties, right? And that's what we see with other viruses. They all have, icosahedral viruses have a lot of things in common, and they converged on that right here. And they say mutating the mRNA initially destabilized the protein. And that was the key to getting this evolution, destabilizing changes in the protein. So, you have an initial 60 subunit, very stable structure. You have to get away from that. Otherwise, it's not going to change because that's pretty stable. So you have to ask it to do other things.
Right.
So 240 subunit capsid, multiple RNA packaging motifs. It's kind of a window on what might have happened early on, right, in evolution, except not at the single protein stage, but when that protein already was able to form a shell, then maybe that's at the stage at which the RNA, or who knows, maybe it started binding earlier. I don't know.
Yeah. I thought this was really fascinating, and I love the ways that it sort of makes me think about viral evolution and how exactly, we evolved, and in fact, how important some of these structures are, given that we are seeing convergence to these same structures here. But I also was reading this thinking about how much I would love to see them take these capsids that they're making, or capsid-like molecules filled with RNA, and I want to know if they can get into a cell.
Yeah. Sure, they could easily. They could add motifs to the protein so that they could interact with. They could stick an antibody on if they wanted to. That would get it into a cell. But they could put a motif on. I'll bet that would get it into a cell.
Yeah, because sure. I was thinking about how this could be really cool instead of using the lipids like we do for mRNAs as an alternative delivery method for nucleic acid based mRNAs or other therapeutics. So not only is this just way cool for evolution.
Yes.
I could imagine some cool ways to use these too.
They do say that they're clearly interested in that, right? They say these may be good alternatives to viruses for gene delivery and vaccine applications, right? People are definitely going to push this forward. Yeah, for sure. Because, virus-like particles are really good vaccines. And so that's what they've made here. They've made a virus because there's a nucleic acid in it, right?
Yeah.
It's not empty.
And an advantage of using something like this as opposed to taking a naturally occurring virus and modifying it down to something like this is going to come without the baggage of prior immunity or having been built for a different purpose or any of that. You can evolve or construct a capsid like this that is specifically for the mRNA you want to deliver.
And it also might get around some types of innate immune responses or things like that might come with some other delivery methods.
They say in the end, this is a cool sentence, the conversion of an enzyme into a nucleocapsid that packages its own mRNA with high efficiency and selectivity shows how primordial self-replicators could have recruited host proteins for virion formation. Now remember, it's all accidental. The RNA bumps into a protein, and if it sticks, it makes it more fit, and it goes on. Yep. It's just very cool.
And it's easy to hypothesize how some of the stuff that they elided here, by engineering it, could arise naturally. So they started, in this case, they started with a protein that naturally forms these 6-dimers, but doesn't have any RNA binding capacity. You could totally imagine in a more RNA dominant world that there could be proteins like that do have the capacity to bind RNA for their normal activity.
Sure.
And then you've got past that step, and then your RNA comes in and takes advantage of that.
Yeah. No, a lot of possibilities. And I think this is a good paper for an evolution lecture, right?
Absolutely.
Because it really fills in some of those gaps there.
Yeah, I was thinking that it could be a good paper for a structure lecture, actually.
That too, yeah.
To talk about the importance of some of these structural features.
Yeah, and you can see when you select for certain properties, this is what you get.
Yep.
You don't have to take our word that all the viruses that are built this way, you have this symmetry here. Look. So some of these people are in Zurich, where I'm supposedly going next year. So maybe I'll go by and do a pod with some of them. I think that would be fun.
That would be cool.
And beautiful pictures, beautiful stuff. I love it. Very nice stuff. What is the boosterism? What was the thing you said? Evolving boosterism.
Yes.
Wow. No one will listen because they won't know what it means, right? All right, let's do some email. Brianne, can you take the first one?
Sure. Karen writes, referencing TWiV 776. Your insurance company will not want to pay an additional administrative fee for a vaccine top-off, not FDA approved. Expect a claim denial if your J&J was run through your insurance and a new bill in the mail from the pharmacy. You might get away with it at a county or state public health vaccination clinic if you claim no insurance. Kaiser Health News, KHN, has documented multiple cases of patients getting surprise billed for administrative expenses by clinics and labs who thought they were getting quote unquote free testing. I actually fit in that category myself, so I have firsthand experience. Don't go to a business and expect something for free despite what the news says. That's from Karen from Yuba City, California. And so this is in reference to our conversations about people who had a J&J vaccine wanting to get a booster of Moderna or Pfizer.
Yep.
And I think that's exactly right, that you are probably going to show up in the system as having already used your insurance, and your insurance will then bill you.
Not only are you on your own medically and scientifically, you are on your own financially.
Yeah, I don't think you need to do that, as J&J is a good vaccine.
Yeah.
And don't do it. But that's a good point. I hadn't thought about that. Thank you, Karen. Alan?
Grant writes, Dear TWiV, as a pathologist and TWiV-herian, person who listens to TWiV, I have to deal daily with incorrect information about COVID-19. There is a plethora of pseudo-experts disseminating incorrect information or opinions, and I now label this syndrome miscoviditis. Wearing masks has proven to be fundamental in stopping the spread of SARS-CoV-2 virus and COVID-19. Masks won't, unfortunately, stop spread of miscoviditis, but gagging would, if only that could be done. Keep up the conversation. And Grant is a pathologist in Perth.
And Grant, ticks all the right boxes. He's got the spread of SARS-CoV-2 and COVID-19. Excellent.
Very good.
Excellent, Grant. Charles writes, another hot and humid day in Chapel Hill, 85F, 29C at 6 p.m. Looks like the weather will be about the same in New Jersey this weekend as in North Carolina. I hope to get a nice view from Mount Mitchell, New Jersey, not North Carolina, or the Twin Lights State Historic Site this weekend. I have no idea what any of those are. And I live here. Shame on me, right? No virus questions this time. Just wanted to say that the shows over the last month or so have been really good. They're close to the back-to-back wonders of 640 and 641. Was that Minna and Fauci, maybe?
Maybe.
I think was.
One of those is Minna, yes.
Would like to share a couple of quotes from religious sources. The first is derived from a Gautama Buddha quote, 3 things cannot be long hidden, the sun, the moon, and the truth. I really hope that is correct. We have had way too many people in high places of power hiding the truth, and it has and continues to cost us dearly. Second is from Stan Rushworth, quote, instead of thinking that I am born with rights, I choose to think that I was born with obligations to serve past, present, and future generations and the planet herself, end quote. Pandemic has turned a very bright spotlight on rights and obligations. Rights without obligations seem to have won out, leaving all of us in much worse shape. I hope this can change. One more quote that really just blew my mind from Ohio State President Christina Johnson, quote, we decided together that we're going to look at this not through a political lens, we're going to look at it through a public health lens, end quote. And he provides a link for that statement. What she was looking at was whether to encourage or require vaccinations for students. Their decision to not require vaccinations showed that she did look at the problem through the political lens, not the public health lens. Drives me a bit nuts.
Yes.
Oh, that's so unfortunate. How could you look at it through a public health lens and decide not to vaccinate your students? This is from Ohio State. Yep. One last thing, Dr. Racaniello, I wish your dog a speedy recovery. Thanks, Charles. So an update on the dog.
Yeah, how is your dog?
Luna. So I talked about it on a Friday, right?
Yeah, last
Friday. It was on Friday.
On Saturday, my son and wife took her to a different vet who'd been to before. Now, the vet experience is really weird because you can't go in, even though you can't. You have to sit in the car. They come and get your dog. They bring it in, and then they call you. And that's just so weird, right?
Just had this experience with both of our cats. They had to go for their regular checkups. And they come out to the car, you hand the cat case over to them, and then you sit there in your car waiting, and they call you.
So they said, it's not Guillain-Barré. They said, no, it can't be. So my wife is like, I know what this is. My husband's a famous virologist. He knows, which kind of embarrassed me. They said, no, that only happens within a day, which is not true. It can happen within weeks. They said, well, we think she has a cervical something, slip disc or something. Let's bring her in Monday for an MRI, where you have to bring her and leave her there overnight, and they sedate her. And my wife said, why don't we medicate her for a few days and then see what happens? So they gave her prednisone and gabapentin. So our other dog had taken gabapentin, which is for neuropathy.
Calm down her immune system and calm down the dog.
Yeah. And after 3 days, she was back to normal. And now she's eating, her appetite's back, her personality is back, her tail is up.
Oh, good.
She's leaping all over. Although that vet said, try and not let her jump long distances. It's not really a good idea. But my wife said she kind of yelped the other day when she jumped. So maybe there is something underlying. So she seems okay, but I just don't know if there's something there. And we should give her an MRI. The problem is the last time she had general, she almost died, right? They had to stop the general. She was having some tooth work and her pulse started to drop. So they stopped. And, the worst thing is to have them take the dog away, and then the next day they call you and say, sorry, I don't want to.
Yeah, that would be awful.
Bad.
Be kind of crappy. So we're going to wait a bit and see what happens. But we're going to give her the course of the prednisone and the gabapentin and see what happens. So that's the story. Thank you, everyone. A lot of people have written. I appreciate it. I thought, as I posted, I said, maybe I should have cut that out because people are going to say, I don't want to hear about your bloody dog.
Now the show's going to be all about the dog. No, that was good.
But people seem to be okay. So
well, I'm glad she's recovering.
There you go.
Yeah, she's better.
Brianne, you're next.
You picked me to read this one just because everyone thinks I don't swear, right?
I thought I knew you did, right? Yeah.
So Jeannie writes, hi all, the Delta variant. It may be outrageous to call it more contagious, but oh shit, is it more fit? Thanks for your show, Jeannie.
That's funny. I really like that. I think our listeners are getting it. There's a report out from Public Health England this morning, which says that now the secondary infection rate of Delta is going back down, which makes me say, really? So you really think it's inherently biological, more transmissible? If it's changing, it's human behavior that's changing. What we've been saying all along.
It's almost like some people are having some immunity to it, so it doesn't transmit as well.
Or that maybe you're doing more secondary attack rate studies, and so you're converging toward the mean for the virus.
That's cute. That's a cute poem. I like it. Very good.
That's
Alan.
A good poem. Yes.
Alan.
Okay, Bonnie writes, Dear TWiV team, when listening to episode 776 and the study on infecting Aedes mosquitoes with Wolbachia pipientis was discussed as a method for decreasing dengue virus transmission, I was reminded vaguely about a short story by Ray Bradbury in which a person travels back in time and causes the destruction of a butterfly, which leads to a change in an election result eons later or some such. Let me explain my concerns. As a fellow in infectious diseases at Washington University, I was fortunate enough to work in Dr. Gary Wiles' lab studying filariasis. Human filariasis is spread by mosquitoes, usually of the Culex genus, and causes horrible disfiguring disease, edema, and death. The primary parasites in humans in this group, as TWiP fans may already know, are Wuchereria bancrofti and Brugia malaya. Interestingly, a major treatment to decrease parasite burden for these terrible diseases is to kill the Wolbachia endosymbionts, which can greatly reduce disease burden of both the adult and filariform parasites. Doxycycline has been effectively used to decrease Wolbachia and therefore worm burden. Treating these parasitic infections with ivermectin or albendazole alone requires years of treatment, up to 15 years or more, which makes eradication a major global challenge. There's even an organization called AWOL, the Anti-Wolbachia Consortium, which has been studying antibiotic treatments against Wolbachia to enhance the elimination of filarial parasites from human hosts. So the Wolbachia is infecting the mosquito?
No, the parasite.
Is infecting the parasite.
And it's required
this thing really gets around.
For parasite fitness.
Oh, yeah. Right. It helps out the parasite. Okay, closer to the home front, one of our rescue dogs required treatment for heartworm due to the canine filarial parasite, Dirofilaria immitis. Part of standard of care now is to include doxycycline to kill the Wolbachia prior to the anti-parasite meds, in addition to steroids to decrease inflammation. Much of the disease process in canine heartworm infections comes from release of WSP, Wolbachia surface proteins, which leads to inflammation, pulmonary scarring, etc. I have unpleasant lab memories about extracting these parasites from dogs' lungs, which had succumbed to heartworm for use to extract antigens, etc., for some of our research on animal and human filariasis. Onchocerca is another filarial parasite whose vector is the Simulium blackfly, which also has Wolbachia as necessary endosymbiont. This is the pathogen responsible for river blindness, a scourge which the world is having some success in eliminating. Blindness from corneal scarring, etc., associated with this horrible infection is prevented in animal models by eradicating the Wolbachia. Heck, this Wolbachia criteria could even involve your immune podcast as well, as its lipoprotein inhibits host degranulation of his eosinophils and impedes the neutrophil response of the host in trying to eliminate the filarial parasite. While I've treated many patients with the entire spectrum of dengue disease and live in Texas, where the Aedes mosquitoes are quite at home, I am less than enthusiastic about a treatment which enhances mosquito capabilities as hosts for these other formidable pathogens of both humans and canines. While human filariasis is not an endemic concern in the U.S., there are too many unknowns, and I would likely be among the local residents saying, now, wait a minute, if such studies were proposed here. Thanks for the invigorating podcasts. And Bonnie is an MD in infectious disease in Dallas. Interesting points. Different mosquito, and the Wolbachia in the mosquito getting into the filaria. I just don't know.
Yeah.
Is that
seems unlikely?
Is that a risk we should worry about with a different species of mosquito?
I think not, right?
I don't know.
I would think these risks would have been considered when they're looking at the release, right? Because we have ecologists who should know about this, right? So the Aedes is going to deliver Wolbachia to these worms and parasites, but they already have them. So I'm not sure what difference it would make. But I mean, these are all interesting things to hear anyway.
Would it influence the ability of the Aedes to carry the parasites?
Oh, the parasites would infect mosquitoes then? I'm not sure that can happen.
Well, because she's saying that at least in the case of the filariasis, it's being carried by the Culex mosquito. And so if you start having Aedes with Wolbachia, I think that's part of what she's getting at here, is that will this enhance mosquito capabilities to be hosts for these other pathogens?
But if the parasite is carrying the Wolbachia, then it shouldn't matter whether the mosquito has it, right?
Yeah,
that's why I asked. I was confused about whether we were talking about mosquitoes infected with Wolbachia here, because I thought it was the parasite that,
Yeah, I am unfortunately not familiar with these systems at all.
Yeah.
So
I vaguely
I am learning
know
lots from this letter.
This is a very interesting letter.
We need Dixon here.
We need
Dixon.
Yes.
Dixon or Daniel or both. That would help. All right. Here's from Monica. Hello, TWiV team from beautiful Victoria, BC, Canada. It's currently 20 C sunny, not a cloud in the sky here. And I will not have to officially mow my lawn until October again, as there is no sign of rain until the fall. First, I want to say thank you for the great show content and have been a subscriber since the beginning of the pandemic in April of 2020. I'm a physician assistant in the Canadian military and initially stumbled across your podcast whilst searching for science-based information to help me understand what was happening in the world instead of listening to sensationalized news broadcasts. You delivered in spades, and I have referenced your show so often when talking to my patients, friends, colleagues, family. I've lost track. Thank you. However, I have to burst Brienne's bubble. 50-foot mosquitoes do exist. And
Yikes.
Monica sends a picture. The mosquito capital of Canada, which you can find at mysteriesofcanada.com. You can still visit Victoria. We don't have mosquitoes here. All the best, and I look forward to many episodes. This is a sculpture of a mosquito, of course, but it is big.
Yes.
It is.
We said, what if Wolbachia makes 50-foot mosquitoes? And Brienne said, as long as they don't bite me.
Exactly.
Okay, Brienne.
Sure. Anonymous writes, and Anonymous gives a link. I was listening to your episode of TWiV 776 and thought of what they are doing in Key West with mosquitoes to combat disease. I have written many emails in my head since April 2020 when I discovered you. I have enjoyed this podcast and also Immune and the Evolution of Viruses one. I do everything on my smartphone, so please excuse the lack of specificity. Also, please excuse any odd word errors due to using a speech recognition app. I am not a scientist, but you helped save my sanity during the pandemic, so many thanks. In spring of 2020, I encountered the guy who lived next to me in the building in the communal laundry room. He was working on a vaccine for Zika. I checked with him to see if the podcast was good science, since I didn't know enough to be sure. I have lifted many more bound science journals than I have ever read. I couldn't understand everything he said to me across the roar of the machines and physically distancing on opposite ends of the room while wearing masks. He knew about it and said that Vincent Racaniello was the first to do something in particular. I couldn't hear what it was, but that told me I had made a good choice of what to listen to. The temperature has changed many times since I have been listening. Gratefully and anonymously, many thanks.
This is an article about...
This is Oxitec's gene drive technology, which they're using, which is it's completely different from the Wolbachia approach, but it does involve releasing wild mosquitoes or releasing live mosquitoes to do something called the sterile insect technique. And this is to eliminate a vector completely.
Right.
Yes.
And this is being done in the Keys, in the Florida Keys,
Yes.
right? Yeah. It's really interesting. Yeah.
Yeah. All very cool.
I think Google also has a mosquito release program, right?
I believe so.
Right.
I've read about
And they
that.
have trucks that drive around and release them. Yeah, we have trucks.
That's different than having bugs in your software, though.
Yes.
We still have those, though, right? Alan.
Let's see. We are up to Jamie.
Yep, last one.
Here's a plan for naming viruses. Transfer to pathogenic viruses the names of disgraced individuals or families who previously had statues, buildings, roads, etc. named after them. You could have Cox-Sackler virus after the Sackler name that was recently removed from my alma mater, Tufts. Jeffrey Epstein-Barr virus. Ooh, that actually might take care of 2 in 1. Robert E. lymphocytic choriomeningitis virus, Jefferson Davis virus, and for you, Vince, polkiovirus after Leonidas Polk. Shall we contact the ICTV?
Absolutely. Absolutely.
I love these. And Jamie is an associate teaching professor in biology at Northeastern. Those are excellent.
And a long-term listener of TWiV, I believe, Jamie, I've met at BU. I met you at BU. Now you're at Northeastern.
Wow.
Maybe I met you at Northeastern. Maybe you moved. I don't know, Jamie. Thanks for listening. And that's so
Thank
cool.
you.
I love it.
Those are excellent.
Those are really funny.
We could get lots of virus names because there are lots of...
There are lots of people you'd want to name viruses after. That's not very nice to the viruses.
Purdue would be one of them, right?
That was the pharma company for the Sacklers, yeah.
Oh, that's the Sackler? Okay.
And it might be confused with Purdue University.
Yeah, we don't want to do that.
You'd have to be careful with people with overlapping names.
Yeah. Contact Jens Kuhn. Jens, what do you think?
Yes.
Jefferson bis Davis virus. Excellent, Jamie. Great idea. I love it. All right. On that note, it's time to do some picks. Brianne, what do you have for us?
So I have an article that is basically talking about some cooking, but it's an experiment that I've always wanted to do and have never actually done that they write about, and this is called The Science of Chocolate Chip Cookies. And so here they talk about what will happen if you change amounts or some of the details of some of your ingredients in your chocolate chip cookies. So if you add more or less butter, if you add cream, if you add more or less sugar, if you dissolve the sugar, how you treat the butter, all of those things, and how that will impact your chocolate chip cookies. If you use egg yolks, egg whites, all of those good things. And so here they basically do all of those experiments and then talk about what kind of change you get to your chocolate chip cookie and how you change the cookie. And like I said, I've always wanted to do this. And so I love reading through these details.
Kenji Lopez Alt.
Kenji Lopez Alt.
This is awesome.
Who's the science chef, right?
Yes. His dad is an immunologist.
So his dad and I were postdocs together at David Baltimore's lab for a couple of years. And Fred came here for his first job. I don't know. Was he here when you were here, Alan? Fred Alt?
Yeah. I remember Fred. He had a big lab that took up the entire
sixth floor.
That's right.
Yep.
And then he ended up at Harvard.
Yeah, he left
where he was one of my professors in graduate school.
Oh, okay. Yeah. I always liked Fred. He's a very smart guy, had great ideas, and was into eating and other things as well. He was fun. He was a lot of fun.
So I'm living vicariously through Kenji because I don't want to have the batches and batches of chocolate chip cookies that would result from this in my house.
The intro to this is great. My wife said, "Stop making cookies." Yeah.
We have picked Kenji Lopez things before. You probably have, Brianne.
So.
And
yeah.
I remember I heard a podcast with him where he's talking about opening a restaurant. And he opened a sausage restaurant somewhere in California. And he said, don't open a restaurant. It's just very hard to do. He's like...
Restaurants are the most commonly opened business and the most commonly closed business.
Yeah.
They're like everybody thinks they can start a restaurant and very few of them actually can.
Alan, what do you have for us?
I have an article from PNAS just a little while ago, back in June. It's a news feature, so not a research article, but it's got a lot of references to research that I think are very interesting on an important topic. I think anybody who teaches, especially at the undergraduate level, really needs to read this. It's called Keeping Black Students in STEM. And this addresses an ongoing issue that's been an issue for decades. There's been a lot of talk over the years of how do we get more diversity in science and technology fields. And it's an important question because if you've got a bunch of white dudes sitting around a table, they're going to have a constrained set of ideas, and that's not what you want, especially in science and technical fields. And so there for years, there have been these recruitment efforts, try and get more students into these fields. And they've had some success. But the problem is that those students, especially Black students, tend to leave those fields. Mm-hmm. What's going on? And we tried to recruit them and they didn't stay. And this article dives into that and explains some of the causes that have been worked out and some of the ways to approach this and solutions. And I think it's really just important reading.
I'm looking forward to reading it.
Yeah. Very good. Yeah, I mean, this is not a problem we're going to solve overnight, and there are a lot of issues that need to be seriously addressed, so this is good.
And this is not an article that's going to say, oh, if you do this, this, and this, your Black students will stay enrolled in it. No, it's not going to be that simple, but it's a whole systemic series of changes and things that we need to think about as we progress with this.
As you said, faculty diversity is important, right? You can't have models if they're not in the room, right?
Yep.
So that has to...
You can't be what you can't see is the saying, I think.
My pick is a new program announced by NIH, Antiviral Program for Pandemics. Why didn't you do this 5 years ago?
Well, I can tell you some reasons why it wasn't done 5 years ago. Let's not
But
go
it
there.
could have been, and maybe
Of
we would have
course.
avoided this. Anyway, the antiviral program developed antivirals, not just for SARS-CoV-2, but to make a pipeline against other viruses with pandemic potential. And you know who they are besides coronaviruses, the paramyxos, Hendra, Nipah, et cetera, Bunya viruses. Picornaviridae is on this, but apparently that's hepatitis A virus only. Filoviruses, Ebola virus, Toga viruses, flaviviruses. So this is good, right? And there's an accompanying article also from the NIH, HHS, Biden administration invests $3 billion from the rescue plan as part of this COVID-19 antiviral strategy. So that's great. I just would like to know how this is going to be done, right? What are you going to do? Are you going to ask for proposals? You're going to do it all within the NIH? What are you going to do?
It's going through, so it's going through BARDA,
BARDA,
NIAID. And so it's pretty much the NIH funding mechanism. So there will be calls for grant proposals. And I'm sure people who are working in this field already are going to be looking at that.
They should. I think it's a good idea.
Absolutely.
But you need to make sure it gets to candidates that can get to phase 1 and be ready.
You have to get them past the valley of death. You have to get, and that's why $3 billion, I mean, that's a significant amount. This is not just a few R01s. This is the kind of money that you would need to do some clinical trials, because that's the really expensive part. Anybody can find potential leads that maybe work in mice. Well, not anybody, but you get my point. That is that is going to be the relatively cheap part of the process. Then you need to get at least through safety trials on them, and look at some human data.
By the way,
so they're actually ready for use.
Yes.
Anyway, so this is good news. I think that's a good thing to do, even though it should have been done. All right, at least we're doing it now.
Yeah.
And Daniel said last night that this week we should have some exciting news about an antiviral. I'm guessing it's molnupiravir. Maybe the results are coming out. So cool. That's the orally available one.
That's cool.
Which he thinks would be great for the fall because it should stop transmission if there are any outbreaks. So that'll be good. And of course, within weeks, we'll have resistance and that'll be the end of that. But, at least.
And you'd also have to deploy it quickly enough and do some kind of a ring containment strategy. So you still need to test and trace and isolate.
TTI.
Yep.
What did Kathy call it?
Tetris.
My
Testing,
God, that
tracing, and isolation.
seems like so long ago. I don't even remember. Oh my gosh. By the way, Alan, I had a pick which you probably liked last week.
I heard it last week. I said, I totally. Do
you know that channel?
I have my subscription list on YouTube because the app is kind of annoying with things you subscribe to. But I have definitely watched a number of their videos, and I know quite a few flight instructors use them for instructional purposes.
Like, cool.
You want to see a good briefing on approach. Watch this, and it's because it's airline cockpits, and they're doing everything by the book.
Very cool. I watched one this morning, a lady piloting an airplane into Mexico City, and she couldn't be more casual. I mean, they do
this every day.
Holy crap. She's just gear down, blah, 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 this autopilot, blah, blah, blah. She's going back and she's looking around and the thing, this big mother plane is in the middle of the city with all these houses and she just, and then boom, it comes down. It doesn't even look like she's working very hard, right?
No.
Are these things auto landing basically?
No, they're not. Well, okay. So there is auto land, which is available in the most advanced airliners.
It's on Route 22. You can buy any car there.
right. Come down to auto land. We lost our lease. No. There's a very advanced instrument landing system that has at the best equipped airports. So if you're flying it in Newark and it's fog all the way down to the ground and they're landing the plane, they're probably using autoland, but it's only the latest, most advanced aircraft that can even do that. And the crew needs special training. And the training for the crew is basically all about what do you do when it fails at this point? What do you do when it fails at this point? What do you do? And it's just putting them in the simulator and failing things at various points because it's a finicky system. But for most of the landing, in most airlines, they're going to have autopilot at least tracking the approach course, and then at the very the end of the approach, the pilot is going to take over and land by hand, partly to keep in practice for when the system fails, but also because that's the point where the system is having the hardest time keeping things precise, and a human can visually do that much more easily.
Yeah, it was cool. The first,
but officer, they are the controls are all boosted on the big airliners. So the planes I fly, you've got basically wires.
Yeah. So the auto goes down to a certain altitude, and then you take over, right?
Yeah, it depends on the aircraft. It depends on the sophistication of the autopilot. If you're flying in a small regional turboprop that's going between Caribbean islands, it's probably a much less sophisticated aircraft with the autopilot. But certainly in the planes that are featured on that channel, like the big 777s and the big Airbuses, the autopilot handles the altitude changes and everything, and it does
turns too, right?
Stuff.
Does the
turns?
turning?
Oh yeah. Oh, the turns are the basic thing. That's yeah.
So don't pilots lose ability to do all this stuff?
That is an ongoing discussion, and there's a whole lot of concern, and there are a number of cases. If you read the NASA Aviation Safety Reporting System, the reason these don't make smoking holes in the ground is because the crews have been trained, and they're got a couple of people in the cockpit, so that things deteriorate and they that didn't go well, and maybe they bust through an assigned altitude or something, but nothing bad happens. They manage to recover because there's enough redundancy in the system. But there's this whole phenomenon that I think some of the self-driving car companies are just starting to realize, like, oh, wow, isn't that interesting? I wonder if anybody knew that. Yeah, the whole aviation field's been talking about it for years. That when things are on autopilot, and then suddenly the autopilot disengages for some reason, there's something that's it's called automation shock or autopilot shock, where there's an orienting response, even by the best trained pilots, even if they were anticipating the possibility of autopilot disconnecting. They may do wrong control inputs initially. They may have a hard time getting a grasp on what exactly is happening. And that's a real problem. Yeah.
No, these are fascinating because I've never been in one of those. The best I do is I look in when I walk by and see all the lights, It's just very impressive. So to have a couple of cameras in there is pretty cool. But I haven't yet seen them doing
well, on the Airbus, you're not going to see that because the stick is down at their side. They're basically sitting in an easy chair with a stick next to them, and that's controlling the aircraft. In a Boeing, you get a yoke.
Yeah.
And you might be able to see the yoke moving, but it's not going to be especially dramatic.
So small motions make enough of
small motions, right? And it's not a big muscle activity. It's just you got to do the right things at the right time.
I enjoy them. They're a lot of fun. The one into Mexico City, the first officer was calling out the altitude, right? And then at the end, it goes really quickly.
Yep.
And all of a sudden, boom.
Yeah, you got to do that on approaches. You got to call out the altitude you're going to and the one you're coming out of. And
the thing on takeoff, which is really cool, right? They have the speed where
V speeds,
three different V speeds, right? And before they go, okay, here's VE 1, 2, and 3. So 1 is when you pull up, and 3 you can't go back anymore. Is that right?
So there are speeds where, welcome to This Week in Aviation.
This is the last one. Okay.
Where a private pilot and two civilians talk about, so the speeds are different responses that you would have if something goes wrong. Aviation, there's this, as you figured out by this conversation, there's this huge focus on what do we do if it fails here? What do we do?
Sure, you should.
And takeoff is the worst time to have something go wrong. So you have to plan out, what am I going to do? I even do this in the little airplanes that I fly. And you are about to take off, and you say, okay, if I have a problem before I get to the last taxi, before I get to the midpoint of the runway where that taxiway is, then I'll stop straight ahead on the ground. And if I have a problem after I've lifted off, then but I'm below 100 feet, I'm going to land in that field at the end of the runway. And above 100 feet, this is what I'm going to do. So those V speeds are cue points for what they're going to do. There's a speed beyond which they can't stop in the remaining runway length. And that's got to be calculated for each flight based on the weight of the plane and the length of the runway. And so that's why they go through those every time, because they're going to be different numbers every time.
Yeah.
And then there's a speed at which, they would return to the airport or turn around or do this or that. Or
I see why you like flying, because it's just
it's these interlocking systems that are so well engineered and evolved, and it's very cool. Yeah.
So I took some flying lessons in college. I even soloed, but I stopped. It was just too expensive. But I had one experience. I was in Ithaca. I used to fly out of Ithaca Airport. So my instructor, she said, there's some big planes here, and you have to see if you're landing and one is taking off. You have to, I don't remember what she said. You have to land in front of where they took off or behind, because there's a lot of turbulence. And I remember once I was alone and a big plane took off and I go, shit, what did she say? I don't remember. I couldn't remember if it was before or after. Anyway, it was okay. But
wait a couple of minutes is the solution.
I said, I don't think I should
turbulence
fly. I don't think I should fly anymore. That's enough of that. But it was very pretty flying up there with all the farms.
Oh, yeah.
And she even, in the early lesson, she flew over Watkins Glen. She said, hey, there's the racetrack. Cool. All right. It was fun, but too expensive. Yeah. We have a listener pick that's right up an alley for this discussion. This is from Mark. Dear TWiV team, at 0:45 hours, BKK reports 89F, 27C, 90% humidity, wind 100 at 4 KT. What is that? What is that, Alan?
Where is BKK? Is that in the U.S.?
What is it, a radio or something?
So that would be the airport code.
Oh, I see.
I'm not finding it. I don't know this one.
Right.
There is Bangkok International.
Maybe that's it, because
BKK.
he's got a link for you, the ThaiFlyingClub.com.
Ah, yes. Okay. That's probably what this is.
Also does seem like 90% humidity.
Yes. Okay. Based on the weather report, this is probably,
So Mark has some more aviation picks. He has Museum of Flight Curator on the Loose, which is a YouTube video, and then Classic Airliners and Vintage Pop Culture. Flying Men, Flying Machines, a 1960s CIA documentary about Air America in Laos. And then for Alan Dove, the Thai Flying Club, when it opens again. Oh, that's in Thailand. You need to go there, Alan. Right?
Oh, cool.
Yeah. Sriracha, Thailand. Look at that. Very cool.
Yeah.
Little planes. Yeah. And
That looks fun.
Dirt runways.
Yep. Cool.
All right. Last 20 minutes of aviation, but I'm interested. I find it fascinating that somebody can make this huge mother airplane work with all these people, and most of the time it's safe. It's just, and they seem casual as hell.
It's not even just most of the time. It is the safest form of transportation humans have ever routinely used. It's the safety record in airline aviation is astonishing.
Yeah, I mean, and then they're casual as hell, right? They're just
well, because they do this every day. They're gonna do it again, possibly in an hour after the plane reloads.
Yeah, but not all of them have done it as much as others. Some of them are more novices than others.
More experienced than others, but all the ones who are in the cockpit, they've sat for hours and hours in simulated flights, and they've been through the whole aviation. They've got hundreds of hours of experience, or in the US, over 1,000 hours of experience, because that's now the requirement.
Yeah.
Right. Well, and given there's this focus on safety, they also kind of have a checklist.
Yep.
It's not like they have to really wonder about what they're going to do.
That's checklist plural. Yeah.
Yes.
Checklist is cool. Yeah. One of them, there was an iPad on the dashboard. Right.
Everybody flies with an iPad now.
But one guy, he had a little desk. It was a pilot, and one I looked at. That was the Irish Aer Lingus. She was writing stuff. She was calling out stuff, and he's saying, sorry, we have a lot of paperwork still. Yeah. So there's still paper.
Still paper. But the iPad, the standard app everybody's converged on is called ForeFlight. It's used for everything from those Airbuses to the little Pipers that I fly. And I've got my iPad. I've got a little attachment to the yoke for it, even in the tiny little cockpit. And
wow, cool.
Keep my paperwork on it.
All right. Enough of aviation, folks. I understand. I kind of like it. Alan does. And Brianne is being patient. Thank you.
She's going along for the ride. Yeah. Thank you, Brianne.
Well, I'm just thinking about where we're going to go in the plane, and if I can get some good Thai food there.
That's good. Yes.
Thai food sounds good, but you don't have to fly that far. There's probably close to you.
No, there's good Thai food in Madison.
New Jersey. Yeah.
But I haven't been on a plane in over a year.
Yeah, I haven't either.
February
I haven't been on a
2020.
commercial airliner in
Yeah.
like 2 years probably.
I'm probably not going to go on one this year either.
I'm going to be going on one in late July,
Where are you going?
Alaska.
Oh, that's pretty far.
Cool.
Yeah.
Very nice. Big mosquitoes there.
It'll be my first time back on a commercial airline in quite a while.
I have a college roommate who's a professor there, and he always said, you have to come and see the 12-inch mosquitoes. I said, nope.
12-inch mosquito.
I don't think they're 12 inches, but.
I'll see if I can see any large ones.
All right, that's TWiV 779. You can find the show notes at microbe.tv/TWiV. Send your questions and comments, twiv@microbe.tv. And if you like what we do, we'd love your support. We're moving into a new studio, the incubator, microbe.tv/contribute. Brianne Barker is at Drew University, BioProf Barker on Twitter. Thanks, Brianne.
Thanks. It was great to be here.
Alan Dove is at alandove.com, Alan Dove on Twitter. Thank you, Alan.
Thank you. It's always a pleasure.
I'm Vincent Racaniello. You can find me at virology.blog. I'd like to thank the American Society for Virology and the American Society for Microbiology for their support of TWiV, and Ronald Jenkins for the music. This episode of TWiV was recorded, edited, and posted by me, Vincent Racaniello. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.